What if a single gene edit could erase a disease forever? For all of human history, our genetic code was a fixed reality. The DNA you were born with dictated your life's biological story. But today, we're learning to rewrite that story. A technology called CRISPR is changing everything. Think of it as a pair of molecular scissors for our DNA. Scientists can now find a single gene, a single mistake in a string of 3 billion letters, and cut it out with surgical precision. This isn't a vague guess. It's a direct edit to the code of life itself. What was once science fiction is now happening in laboratories around the world. The implications are breathtaking. For the first time, we have a tool that can target the root cause of genetic diseases, not just the symptoms. It's a shift from managing illness to potentially eliminating it. Diseases like sickle cell anemia, which arise from one tiny error in the genetic code, could be corrected. Cystic fibrosis, Huntington's disease, and thousands of other inherited disorders that have plagued families for generations might one day be removed from our vocabulary. But the promise of CRISPR extends far beyond just curing rare conditions. This technology could fundamentally reshape our relationship with health. Imagine a future where we can edit our immune cells to be more resistant to viruses like HIV or even the common flu. Treatments could become hyper-personalized, designed specifically for your unique genetic makeup, making them more effective and with fewer side effects. Health could transform from a game of chance into something we can actively design and improve. However, CRISPR is not a magic wand. The technology is still in its infancy, and it comes with significant limitations and risks. Our genome is an incredibly complex system, and changing one part can have unintended consequences elsewhere. An edit meant to fix one problem could accidentally create a new one. These off-target effects are a major concern for scientists. We also don't know the long-term effects of these edits. What happens to a person with edited genes 10, 20, or 50 years down the line? The truth is, we don't have the answers yet. The human blueprint is not something to be edited lightly. This unprecedented power forces us to confront profound ethical questions. If we can correct debilitating diseases, should we? Most would say yes. But where do we draw the line? Is it fair if these life-altering treatments are only available to the wealthy, creating a new form of genetic divide between the haves and have-nots? What about consent? We can't ask an embryo for permission to edit its DNA, yet that decision will affect every cell in its body for the rest of its life, and potentially the lives of its descendants. The debate intensifies when we move from therapy to enhancement. This is the fear that haunts the CRISPR conversation, the rise of designer humans. If we can edit genes to prevent disease, what stops us from editing them for traits we consider desirable? Greater intelligence, enhanced physical strength, a specific eye color. The line between fixing a problem and pursuing perfection is dangerously thin. Opening that door could lead to a future where humanity is split, not by class or creed, but by genetics. A world where some are enhanced and others are left behind as naturals. Recognizing these risks, governments and scientific bodies around the world are grappling with how to proceed. Strict regulations are being proposed and debated. Some countries have banned all germline editing changes that can be passed down to future generations, while others are cautiously allowing research to continue under tight supervision. International summits bring together scientists, ethicists, and policymakers to forge a global consensus, but agreement is far from universal. The scientific community itself is divided, with passionate arguments on all sides. This is not just a scientific challenge, it is a global, societal one. The decisions we make today about gene editing will echo for generations. The children of tomorrow may be born into a world where inherited diseases are a thing of the past. They may live longer, healthier lives, free from genetic predispositions that burden us today. But they may also inherit a world of new inequalities and unforeseen biological consequences. The very definition of what it means to be human is on a table. And the choices we make in this decade could shape the future of our species in ways we can barely imagine. So, this brings us to the ultimate question. It isn't about what CRISPR can do, but what it should do. Just because we have gained the power to rewrite our own biology, does that mean we have the wisdom to use it? Because the future of humanity isn't just being written, it's being edited.